chromosome is a long length of DNA. And on this long length of DNA, there are different sections of DNA which do different things. And the first thing that's important is this piece of DNA at the end, which is called the telomere. And the telomere is a special piece of DNA. It doesn't code for any proteins, but it is really tightly coiled together. It never unravels, and the purpose of the telomere is like the things on the end of your shoelaces. It stops the rest of the DNA or the rest of the chromosome from becoming unraveled. So here's our telomere at the end holding this chromosome together. Then we have our stop. And this tells the cell that there is a gene coming up. Here we have the gene. This is the coded bit of the gene, this is the complement of the gene. And at the moment, that gene is inactive. It's not switched on because it's folded on itself like that. But this piece of DNA down here is called epigenetic DNA. And what it does is it tells this gene when to switch on and when to switch off. It responds to changes in its environment. It doesn't code, it's got no code, it doesn't make proteins, but it's the master switch. So this piece of epigenetic DNA wants this gene to become active. It switches the gene on. The gene literally opens up like that to become active. And then it does exactly what you guys did. It codes. It reads the first codon and says it's that amino acid, and in the cell that amino acid starts to form. It decodes or translates that whole piece of DNA, just like you guys did here, and from that, out in the cytoplasm, a protein is made. Just like that. The epigenetic DNA tells the gene to switch off, so the gene closes and it stops making proteins. It opens and it makes proteins. It closes and it doesn't make proteins. The cell knows where to stop reading this gene because there's a stop down there. So this gene here, a section of DNA that codes for a specific trait, in this case it's coding for this protein, has undergone some mutations. As it's replicated or as it's got old, it has changed some of the codons. And that leads to different versions or different alleles of the gene. This allele produces that protein. This allele produces a protein which is very similar, but it's got some differences. This allele produces a slightly different protein again. Some genes produce different alleles simply as they age. If you go home and look at your thumbnail and compare it to your mother's thumbnail, you'll see that the type of protein being formed by an older person is quite different to the protein being formed by a younger person. And that's because of the changes in the DNA over age. So, going through here, telomere, our stop, closed gene, stop, epigenetic DNA. Another gene opens to produce proteins, closes to not produce proteins. What tells it to open? The epigenetic DNA. And so the story carries on all the way down here. Gene, epigenetic DNA, gene, epigenetic DNA, gene, epigenetic DNA, gene, epigenetic DNA, and so on, right down to a telomere at the other end, holding it all together. Now, you imagine this inside a cell, and it's all loose and floppy. Well, what it needs to do is protect itself. So what it does is, when it's a chromosome, it's actually coiled up as if you take a shoelace and twist it two ways. It curls up and then it starts to knot up. That's what your chromosome is. And we could stand here and do this. We could stand at that end and that end and twist and twist. We'd be here for probably a day to get it into a nice tight chromosome. In your cells, it happens in less than a second. Curls up, opens up, curls up, open up. And that's the story of your chromosome. Okay?